thanks.
Time as any, Mr. Newman. Take her down to periscope depth.
Yes, sir. Clear the bridge. Dive.
Stand right.
Greetings, everybody. I'm Sal. I'm Salvador Cordova. Welcome to Evidence and Reasons, a YouTube channel devoted to studying evidence and reasons for the Christian faith. I'm privileged to be joined by Peter.
Hi.
Hi, everybody. Yes, and we are going to talk about a pretty technical topic today, what I call Muller's limit and the origin of junkDNA theory. Peter had contacted me and was asking questions to try to explain things. I'll do the best I can to explain some topics. And then after I'm kind of done with my slideshow, there might be other questions and we'll just see if we have time to address them. If not, we'll have another show. I'm very grateful Peter has approached me and shown interest. This is our second show on almost specifically population genetic topics. So I'm excited at the prospect of talking about this. It's one of my, it's an interesting topic. I'd say my favorite topic is in the area of cellular and molecular biology, but this is also good. Mm-hmm. So with that, let me share my screen. And this slideshow was prepared for actually another talk that I gave some time ago. And let me make sure that I have everything correct. Apologies here. Peter, what do you see on your screen?
I see the digital Muller's limit and the Lewis paradox. I see it.
Okay, great.
Yeah, everything
fine. So this is relevant to the creation evolution debate. However, the way I'm going to approach this is I will mention when something is a creationist theory. I'll try to point out a creationist theory, but by and large, most of what I'll be trying to describe is just straightforward out of evolutionary literature. I may be using some terms or labels that are not specifically in the literature, and I'll try to identify that. So the first term I'm using is actually Muller's limit, not to be confused with Muller's ratchet. Muller's limit is just a term that's just a more convenient term that I'll be using through this discussion, and I'll define it later. So how all this came about, there was a paper dated January 2016 by a respected evolutionary biologist named Dan Graur, and he was estimating the functionless fraction of the human genome based on junkDNA theory, and he uses Muller's limit for that. And he said something kind of astonishing in that paper. He said, studies have shown that the genome of each human newborn carries 56 to 103 point mutations that are not found in either of the two parental genomes. If 80% of the genome is functional, as trumpeted by Encode Project Consortium 2012, then 45 to 82 deleterious mutations arise per generation. For the human population to maintain its current population size under these conditions, each of us should have on average 3 times 10 to the 19th to 5 times 10 to the 35th children. This is clearly bonkers. If the human genome consists mostly of junk and indifferent DNA, if the vast majority of point mutations are neutral, this absurd situation would not arise. So I'm just going to cover the mathematics for how Dr. Graur arrived at this figure. And this was of interest to creationists because he said this in light of the Encode Project Consortium. He said, if ENCODE is right, then evolution is wrong. And that was in his December 2013 lecture at the Society of Molecular Biology and the SESBE, which is the Spanish version of that society. And let me just read something he had in his slideshow. He said, 3 problems. If the human genome is indeed devoid of junk DNA, as implied by the ENCODE project, then a long, undirected evolutionary process cannot explain the human genome. If, on the other hand, organisms are designed, then all DNA, or as much as possible, is expected to exhibit function. If ENCODE is right, then evolution is wrong. So I'm just going to go through his mathematical reasoning. I'm not immediately going to say he's right or wrong, but I'm just going to try to state the theory and let people decide how close it is to reality. Now, Dr. Graur, again, he is an evolutionary biologist. Mm-hmm. He had this to say about the National Institutes of Health, ENCODE project. He said, they're ignoramuses, crooks, the scientific equivalent of Saddam Hussein, and he called the director a creationist. So I'll pause here a little bit. Are you familiar, Peter, with the ENCODE project? Have you heard about it at the NIH?
I have heard, but I'm familiar not much. Maybe you can explain now in details what is it.
Sure. The ENCODE project is a project that was, so we had the Human Genome Project that was just mapping out all the bases in the human genome. So we have a pretty good idea of what all the nucleotides are, about 3.3 billion base pairs. But we actually don't know what each of the base pairs does. And there has been a dispute whether, is it 2% that really do anything and the rest is just non-functional, or is it 10% that do something and the rest is non-functional, or whatever proportion. And there's just a scientific discussion about that. And the ENCODE Project Consortium was the first of actually several and ongoing projects to try to catalog the function of each section of DNA. And my best estimate, based on financial statements, is that the ENCODE Project Consortium has already spent maybe over $500 million in the research. That's $500 million. That's a lot of money. There have been follow-on follow-on projects like the Roadmap Epigenomic Consortium, which is also ENCODE-like and does kind of the same things, uses very similar experiments. It's also funded by the NIH. It's about a $300 million project, and there are even more coming down the pike. And I'll talk about that a little bit. So this is a big money deal. And Dan Graur did not like their conclusions when they said that they believed that most of the genome is functional. It doesn't agree with junkDNA theory, which Dr. Graur subscribes to. And he's taking on a pretty big project because there are a lot of, with that amount of money and the number of researchers involved, this is one reason he's upset, and that's why he's calling them ignoramuses and crooks, because there's a lot of money involved in this project. He's very upset about this. And this fight is even appearing in universities where we have faculty members at the same university disagreeing with each other. So on the one hand, Joe Felsenstein at University of Washington will argue that against the ENCODE project. And then there is an actual ENCODE researcher, also a faculty member, John Stamatoyiannopoulos, who's also there, and he's arguing ENCODE is right. So even in our academic institutions, there's a line being drawn between evolutionary biologists, population geneticists on one hand, and on the other hand, medical researchers. This is kind of a quiet fight, but it's also kind of interesting that they would be at odds. So this is from Joe Felsenstein's book on population genetics. The name of the book is Evolutionary Theoretical Genetics. And he wrote it in 1970s. In the 1970s, there have been many editions of this book. And I'm quoting from the 2017 edition. Felsenstein writes, "The mutational load calculation continues to be relevant to understanding whether most eukaryotic DNA has any function that is visible to natural selection. Recent announcements, ENCODE Project Consortium 2012, that 80% of human DNA is functional based on finding some transcription or binding of transcription factors in it are very misleading. Junk DNA is still junk DNA. However often its demise has been announced." So that's one viewpoint. And on the other viewpoint, that's Felsenstein's view. The other view is that it is functional. So there is an impasse as of this writing that has not been resolved. And what I mean by resolved is one party will say, hey, you know, I was wrong. That's not happening right now. So I mentioned the ENCODE Project Consortium, and there are other projects, big money projects that are exploring the functionality of the genome, like the 40 nucleome project. That's really exciting, and all these others. So this is a big money deal. And right now the evolutionary biologists are, some of them anyway, are pretty upset that so much money is going into this because it advocates for functionality in the genome. So that is kind of surprising that there is this dispute. There are other projects than this kind of peripheral. So someone I've had the privilege of working for, I still work for this gentleman, but by mutual agreement, the contract may be winding down. I've worked for this gentleman for almost 7 years now. John Sanford, October 18, 2018, he was invited by individuals at the National Institutes of Health. He was able to, even though he's a creationist, he was invited to speak because he is a respected applied geneticist. He invented the gene gun, and so a lot of genetically modified organisms on the planet are through his process. So they, you know, it was thoroughly appropriate, even though he's a creationist, to at least give him an audience. And he was talking about just peer-reviewed literature that talks about deterioration in the human genome. And he cites one of his colleagues at Cornell, and also who was at the NIH at some time in his career, Alexei Kondrashov. Kondrashov wrote this book, The Crumbling Genome, and he says, in the average human genotype, over 100 genes are dysfunctional or altogether missing, and over 1,000 genes are substantially impaired. A newborn human carries about 100 new de novo mutations originated in the germline of their parents, about 10% of which are substantially deleterious. And so this is the figure that, this relates to Graur's figure. If we take, go on.
May I ask,
Yes,
so how does they come to these digits, like 99.9% elimination? This would be interesting.
Yes, I'm going to cover that math today.
Mm-hmm.
So but I'm going to show you the formula first, and then I'll give the proof for it, and I'll show how it is. I'll reflect. I'll derive it from peer-reviewed literature, just so I'll show you. I'll give you the references as we talk, just to reassure you I'm not just making up the derivations. So if we have 100 new mutations, and about 10% are deleterious, meaning they hurt the genome.
Mm-hmm.
All we have to do is to determine how many offspring we have to eliminate, we take E and raise it to the -10th power and do that. And this isn't the best way to state it, and I'll go a little bit more into that. But Dr. Graur was pointing out that if ENCODE is right, we would need E to the 80th power, and that would imply 10 to the 35th offspring. So I'll just go back to his. And there's going to be a little bit of a problem that's like, okay, when is it E to the -80th versus E to the 80th, et cetera? Or when do we put in 1 minus E? I'll try to clarify all that, because in the literature, for those new to it, it'll be kind of a little bit confusing. So we have this figure 5 times 10 to the 35th. That's roughly E to the 80th power. And we can work through the numbers, but I'm going to work through the derivation first, and maybe that will help. I hope it helps. So this, I'm going to focus on how Dr. Graur arrived at about 10 to the 35th offspring. Here it is. So we'll see this E to some power, E to some power. And another way of stating it, which I think is clearer, is if we have a number of bad mutations, how many children would we have to, each person need to each female has to have in order to be able to have at least one offspring that is free of the bad mutation. And this relates to the Poisson distribution. But I'm just going to state the formula, and then I'm going to clarify how it was derived. And the formula is just basically this: 2 times E to the U. So for when Dan Graur was saying 10 to the 35th, the exact number should be 82. And when you raise it, when you raise E to the 82nd power and multiply it by 2, you get 10 to the 35th on that order. And so now I'm going to justify how this formula was derived. So again, I'm just trying to explain how Dr. Graur got this figure way back here in his paper. Let's see if I have it. Mm-hmm. Yeah, this figure here. It's basically, where did he say it? So if we take 82 here, and then we say 2 times E to the 82nd power, it's going to be around 10 to the 35th. He used a slightly different method, and it's within 1 order of magnitude. So now I'm going to show how this was derived, and this leads to junkDNA theory. So when let me just go through this, and hopefully this will start to be clear. So what I was trying to point out is when Dr. Sanford was using these terms here, one will see it in the literature. So this is from a paper by Iyer, Walker, and Keightly. And you could see here this 1 minus E to the minus U, and then they give a citation. So they talk about, like, if you have U, wait, I'm sorry, is this, let me make sure I have the citation correct. I think this is Nachman and Crowell. I'm sorry. This paper by Susan Nachman and a gentleman named Crowell, no, Susan Crowell, Michael Nachman and Susan Crowell, I'm sorry. You'll see this term 1 minus e to the minus u. So if u equals 3, one needs 40 offspring. And you could see how that. So if we have 3, Nachman and Crowell, 1999, and I can give the references. You just plug those figures in. So it took me a while to dig that up. This is, unfortunately, people in the know who are in this community know what these numbers mean. For outsiders, it takes a little bit of uncoiling. And then, yes, this is the paper I just referenced where we see that, Nachman and Crowell. Then we'll see it in another paper, this 1 minus e to the minus u. And why don't I just read it? It has been estimated that there are as many as 100 new mutations in the genome of each individual human. If even a small fraction of these mutations are deleterious and removed by selection, it is difficult to explain how human populations could have survived. If the effects of mutations act in a multiplicative manner, the proportion of individuals that become selectively eliminated from the population, proportion of genetic deaths, is 1 minus e to the minus u, and it gives a reference, where used the deleterious mutation rate per diploid. So high a rate of deleterious mutations, u much greater than 1, is paradoxical in a species with low reproductive rate. So let me see if I have, yes, that's the citation. High genomic deleterious mutation rates in hominids, 1998. And this problem has not gone away. It's gotten worse, and that's why Dan Graur is very upset. So this debate is continuing. And so I'm just laying out the debate. So in this paper, we'll see this citation of Muller's paper called Our Load of Mutations, and it's a 1950s paper. Yes, that's about 70 years ago, but that's a landmark paper. The ideas are basically still intact because it's just population genetics math. He won the Muller won the Nobel Prize in 1946 for his work on mutations. So he's a very respected scientist. So this is what I call Muller's limit. In this paper, Our Load of Mutations, and this is available on the internet for free, he said U is approximately 1.0, the number of mutations per individual per generation. So that's the number of mutations per individual per generation. If it's there or slightly above it, the human genome will deteriorate. So if
yes, so basically you plug it with this formula, n is equal to 2 times
e to the u,
e to the power of u diploid, and then okay, yes, I got it.
Okay, very good, very good. And so see, I told you in before we went on air, I said you're going to get this quicker than most people. And so I hope you'll forbear with me. You may already understand a lot of this. You'll understand it better than most people I've talked to. But for the sake of our listeners, I'm going to go into painful detail. So we'll see this number again. JunkDNA theory was put forward in this paper in 1972 by Susumu Ohno. And he'll make reference to this Muller limit. So this is what he had to say. He said, there seems to be a strict upper limit for the number of gene loci which we can afford to keep in our genome. Consequently, only a fraction of our DNA appears to function as genes. The observations on a number of structural gene loci of man, mice, and other organisms reveal that each locus has a 10 to the -5th per generation probability of sustaining a deleterious mutation. It then follows that the moment we acquire 10 to the 5th loci, the overall deleterious mutation rate per generation becomes 1.0, which appears to represent an unbearably heavy genetic load. And so that was from his paper, So Much Junk DNA in Our Genomes, from the Brookhaven Symposium in Biology in 1972. Now, I'm sorry for kind of the technical language here, and it's a little bit obsolete. This idea of counting gene loci, that's not the prevailing view. But my point in all this is to show that this number of 1.0 came from Muller.
Yes.
And I believe in this paper by Ohno, he will reference Muller's work. So this limit of 1.0, I mean, it's just kind of funny that the number is 1 for humans, that kind of makes it easy to remember. So now we're going to go to some of the math. So I was quoting this paper by Walker and Keightly, and this reference list has 6 references. So I cited one reference here, Muller's Our Load of Mutations. I'm now gonna look at this paper by Kimura and Maruyama. And so that's why I highlighted this. So let's look at this paper, and this will give the mathematics for how the Muller limit is derived. So this is the paper, and this is the formula of interest. This is a statement of the Poisson distribution. It's right here. And you can even see that Kimura uses the word Poisson distribution. That's equation 1.4. So this is, I'm going to explain this, but I'm just trying to point out this is accepted population genetic literature. I didn't just pull this out of the air. This is where it came from. So even though this was 1966, Muller probably laid the groundwork for it, and because it seemed probably obvious, they didn't even probably need to cite this because the Poisson distribution is well accepted for how mutations may be distributed. And we'll cover this, we'll continue to cover this, and I'll explain more. Just as an aside, also in this list, Walker and Keightly's reference list, they cite Lande. It's just an interesting, I'll just cite the title, 1994, The Risk of Population Extinction from Fixation of New Deleterious Mutations. So this all relates to extinction and junkDNA. It also cites this paper by Kondrashov. And Kondrashov says, why have we not died 100 times over? So there's concern that if we have a very functional genome and we have all these, the basic concern is if we have more than, say, per individual, 1 deleterious mutation per individual per live birth, we shouldn't be alive. So this is still an outstanding question in population genetics. So I'm not quoting creationist literature. This is mainstream literature. And he wrote this book. He was a colleague of Dr. Sanford, who is a creationist at Cornell. So going back to Our Load of Mutations. Oh, please. Okay. So we could see it here, this 1.0. And again, this has all this, there's a lot of context here in the meaning of what, how this was derived. But what I'm just trying to point out is, again, this number 1.0 appears even in Muller's original paper. So I'll just read it verbatim. It says, it is interesting to note that the maximum possible value for eliminated individuals could not be more than 5 times this minimum. And this is the minimum here, 0.2. That is, it could hardly be above 1.0 as reckoned by the approximation method that in which correction is not made for overlapping. And it's pretty involved, but again, that number 1.0 appears. Then taking the minimum or most conservative value, 1 in 10 or 0.1 for the total mutation rate, we find the rate of elimination of individuals, or NT, the rate of appearance of new cases, must be at least 1 in 5 or 0.2. So I'm sorry, I'm not going to bother to explain this because it's really involved, but I just wanted to point out that number 1 does keep pointing appearing, and it's going to be moot however I interpret it in light of the way we understand. And so this is, I'm going to kind of show how this can be arrived at in a variety of ways. So if parents by and large have healthy children, and I'm representing it by this green emoji here, and maybe only 20% of their children have a bad mutation. It doesn't have to be a severe birth defect, even a mild one, you know, even a mild one, something that won't kill them. We can eliminate it. And I'm gonna show that with Arnold Schwarzenegger from Terminator 2 or 3. And he's gonna say, "Hasta la vista, baby." And it eliminates he'll eliminate. So he's my metaphor for natural selection. And the population can reproduce. So the survivors are the parents of the next generation. As long as they have excess reproduction, the population can stay healthy. So this works well. If we have just like a 20% mutation and selection equilibrium. The problem happens is if there's 100%. If every child has at least one deleterious mutation, if we apply perfect selection, well, it's going to wipe out the entire population. So, you know, what do we do? There are some proposed mechanisms to solve this problem. And just for completeness, one of them is called soft selection, another synergistic epistasis. I can't cover that in this discussion, and just suffice to say, even these mechanisms are not considered necessarily adequate either, but I laid out the basic problem. Now, there is also a situation where there could be many children that have a deleterious mutation, and one of them happens to be healthy. The problem with this, in this particular scenario where there's 31 offspring with a bad mutation and only one that's healthy, this would imply that there have to be 64 kids per female. This is just, there's a point when the mutation rate is high enough that the females just cannot make enough children. And that's where Dan Graur was saying 10 to the 35th power. So I'm just gonna skip through this. Since you saw, since you, Peter, saw my discussion with Dapper Dino about the definition of fitness, I'm going to skip this. I'll refer the readers to that long discussion debate I had with Dapper Dino and fitness. And it's not immediately relevant to this discussion. If we want to revisit it at some time, we can do that, certainly. So I'm just going to skip that. And yes, this relates to counting how many deleterious mutations there are. Just suffice to say, there's the population genetic approach, which I don't think is valid. It has lots of fancy math, but I just consider it mostly theatrics. I've argued the way to determine a better criteria of fitness is in terms of biological function and structure, but I'm just going to skip that for now. So I think we already kind of, so we do have survival of the fittest, and with the Arnold Schwarzenegger thing, this is just repeating what I said earlier with the pictures of elimination. So I probably should have eliminated this from my slideshow because it's not adding anything to what I already said. So apologies. So this leads to the Poisson distribution. So let's say the average number of mutations per individual is 1.0, which is the Muller limit. All right. So if we have the 1.0, if we use the Poisson distribution, these are approximate numbers because really the Poisson distribution goes out to infinity. I'm only going to go out to 5. So 38.9% of the population has no mutations. If we assume a Poisson distribution, 38.9 has one mutation, 11.1 has two mutations, so on and so forth. And again, once we start to get to the high numbers, these percentages don't work so well because the population model where the Poisson distribution works really well is for an infinite population. So just suffice to say, out here it doesn't work so well. But the point is if there is if there's on average one mutation per individual, all of this on this side here, and I'll make my laser cursor, all of this here will have to be eliminated. And that's when it will start to strain, because this is about 40%, at least 40, 50, over 50% of the population has to be eliminated by selection.
Wait, why? So you eliminate?
If we wanted to eliminate anyone with a deleterious mutation, if we assume the Poisson distribution and there's on average one mutation per individual, if we just wanted to use pure natural selection, we would eliminate all these people that have mutations. So there'd be some people with one mutation, two mutations, 3, 4, 5, etc.
Okay, so you eliminate those. So you let leave only those who has 0 mutations, and other guys are eliminated, like that.
Correct. So we keep 0 mutations. So that doesn't look too bad when the number is just 1, but then it starts to get bad when the number is higher. Oh yeah, so yeah, that's how I visualize it. So we're going to eliminate all these guys here. I mean, 1.0 is not too bad, but that's just right at the limit of reproductive. I mean, there's some point the parents have to make enough children so that we could restore the number of individuals for the next generation. And that's where 1.0 comes.
And may I ask, please?
Yes.
And why do you eliminate all of those and not unless you could let leave like another 83% with one deleterious mutation? Why not that?
That's a very good question. I mean, obviously that could be the case and the population survives. The point is that it will start to accumulate over time. And
how exactly it is accumulated? So let us say so. So now, for example, I let population those units what have 0 mutation and 1 mutation they survive, and other guys are eliminated. So how do you think what mutation would offsprings of them have? How will it be distributed?
Oh, Peter, there's some problem with your audio.
I'm sorry.
I couldn't. There was a lot of. Maybe you're blowing on your microphone or something. There's a lot of distortion.
I'm unexperienced with that. Sorry. So did you got my last question?
I think I'm going to try to repeat it as best as I heard it. So let's say we just eliminate this here.
Yes, 2 and more.
Right.
And
we let this live here. Oops, sorry. I didn't mean to do that. We'll let this guy be included in the survival. And that'll work for a while. But the problem is it starts to remain and be inherited. And it'll start accumulating. That is the theory.
How exactly? Moment. This is a good point. So how do you think what amount of mutation would have these offsprings from if you left 0 and 1 mutation to survive? So let's say it's just roughly.
What rate of mutation would create this situation?
Yes. You do not count new deleterious mutation what will have. You only count those deleterious mutations what are inherited from these groups. This group have survived. So how do you think the numbers would be?
I think it would still be according to I mean, let me say this. They'll survive. I mean, there are plenty of people that have a slight deleterious mutation, like say their eyesight's not as good as someone else. They'll live. It's not lethal. So this doesn't mean they're going to die. What I hope I'm answering your question. It just means that if selection does not eliminate them, that this will be inherited in the population. It'll remain in the population at some level. That's what Muller was concerned with. He said, even if we we can say, all right, let's just assume that selection doesn't work perfectly and it lets these people live. What he was concerned about is that over time, the human genome will accumulate so many problems and defects that the descendants will be sicker than their parents. And I'm sorry, I may not be answering your question. I don't know if, I thank you for watching my discussion with Dapper Dino. So for example,
yes, I watched all your discussion and debate since so. Yes.
Yeah, so there was that one point where I said, okay, is human intelligence declining? Human intelligence. And so there could be a number of factors for that, but that's one example where where maybe a desirable trait like intelligence, it may, the average intelligence may go down. People will still live, but there may be a decline in ability. So it may well be that their mutations in the real world will have mutations, and the children will definitely survive, but over time it starts to affect the genome if it's not eliminated.
Yes, but look, you view only the one generation, yes, count that, and you think that over many generations the amount of mutation will not stabilize, something like that, but it will continuously grow and grow.
Right. That there'll be,
right?
Yes, there'll be one for every generation. If this is what the limit says. If there is hypothetically with each generation one new bad mutation per individual, that is the absolute limit, according to Muller, where if it's gone beyond that, the human race is headed for extinction. That's kind of the simplistic way of stating it. If that mutation rate is sustained at 1.0 or slightly above. And the reason Dr. Graur was weighing into this and this whole fight over ENCODE is the number could be like 80. So 1.0 is like a really small number. I mean, this is just the threshold, and the numbers are like 10, 25, 80. They're just gigantic numbers, and that'll affect the Poisson distribution. And that's why they were.
I understand.
Yeah. This is why they were upset. So that's not immediately creation or evolution. It's just why this dispute came up. And so anyway, I'm going to just show this a little bit, just to clarify the symbols. In the modern literature, we see the U here. That's the mutation rate. In Kimura's paper, or the Wiki Population Genetics, you'll see it look like that, which is more like Kimura's paper. And I'm just going to show how these symbols relate. Okay, so you have the Wiki definition of the Poisson distribution. This is the Kimura version, and this is the modern version. They're the same thing, just different symbols. So this is mostly for the audience that's not familiar with the Poisson distribution. And I'm just going to relate the little pieces here. Just for the sake of completeness, it's the same thing. So I'm just showing where it got derived. So just for those not familiar with the Poisson distribution, when we say I, if I it corresponds to the proportion of individuals with 0 mutations per individual per generation. So if I is equal to 0, it's 0 mutations per individual per generation. I equals 1 corresponds to 1 mutations, and so forth. So again, if I just use this formula here just to show how I got it for this diagram, the Poisson distribution f of u0 gives a number of 38.9%. And so that's how I got these numbers. Again, for finite populations, in these higher number of mutations, it falls apart. So the important one is this one here. This proportion, as the value u goes higher and higher, this percentage goes very, very small till it's like 0.000 some percent. So
yes,
yes, I'm glad that you understand the math, because then we can go through this quicker and I don't have to waste.
I want to ask you a question. May I?
Yes, yes, please.
This calculation, what you point in is it is calculation over 1 generation, right? Have you ever tried to properly view the development of it over many generations with some threshold over maximum amount of mutation possible and so on?
Yes.
Or you just think that you view one distribution over 1 generation and this is enough to make the conclusion, right?
Well, what this is, that's a very good point. So yes, this is over 1 generation. And then I have a graph that shows how it accumulates. I'll show you how this can be extrapolated over many generations.
Okay.
So that's a very good, very, very good question. And I'm sorry if I'm fumbling through some of this. If I don't understand your question, I really do want to try to answer it. So again, we're just revisiting this. So I was just trying to show where all of this came from. And so that's why one will see it in the literature. So some of the questions come is, how come it's 1 minus u, and how come it's sometimes e to the u versus e to the minus u? And there are derivations I could show that deal with that. Let me see if I have it. Yeah, okay. I'm sorry, this is really trivial. We'll just start with this. 1 equals 1, 1 equals 1 plus 0. And we can actually state this. Let me see. Minus e to the minus u and plus e to the minus u equals 0. So this is 0 right here.
Yes. So what is this calculations about?
Yes, I'm trying to relate it to why we see sometimes the term 1 minus e to the minus u, and then sometimes we don't. That's all. That's all. So this is just for completeness, and I won't belabor it. We can do some more derivations. So you'll see this 1 minus e to the minus u term, and it can be transformed. So that's they use this form of the equation here in these papers. And we could say that this represents the proportion of non-survivors. This term here is the proportion of survivors. We can also state it alternatively, if we multiply both sides to e to the u, we can say that's the number of offspring per parent and the number of non-survivors per parent, number of survivors. So I will use e to the u because it's just simpler, but that's how it would relate. That's the only reason I did that derivation. So then e to the u is the total minimum number of offspring per parent. 2 times e to the u is the total minimum number of offspring per female parent. And so going back to the graph at the very beginning of this talk, that's how we finally get just at this number 2 e to the u. So that's just for completeness, just, you know.
Okay.
So now we'll go to the multi-generation thing, and I'll cover it. So let's say we started off with 0, just hypothetically, just purely hypothetically, with a nice perfect genome. And then we had U equal to, say 80 or something. So under, I'll just illustrate this here with a mutation rate of, say, I think I used round numbers of like, 80. So under the Poisson distribution, this would be kind of the extreme. These would be like the 97th percentile, meaning only 3% would be as good as this, because the guys who are really in bad shape are on this end. Most of the guys are here, and a few are, this is the best here. If we had a mutation rate of 80, where U is 80, we could, let's just say that the best people have 60 mutations, and these are gone on to the next generation. So again, this is just one generation, but then we'll repeat the process. We'll, so the parent dies, so the best genome dies just from old age, and we're left with the children that are all have some defect, and we'll eliminate the worst, keep the best, but the best still has 60 mutations. So that's generation 1, and we'll just repeat the process a little bit. Generation 2 is going to add, if we repeated the process, we're just assuming now 60, with an average mutation rate of 80 per individual, but we use high selection. So we add another 60, we still have a problem with the next. And so this process will continue, and I'll just skip a little bit. We'll go to 1 million generations. At 1 million generations, they're just like 60 million mutations. So that's how it accumulates. That was the concern, that at a reasonably high rate of mutations, like say 80, the human population just would not be sustainable. And as I said, the number that has been cited a lot is about 1.0. And so that's where this argument came about, because Dan Graur, the evolutionary biologist, wanted it to be around closer to 1, but the ENCODE Project Consortium is suggesting a number like 80.
So may I ask, where did you get this info about the development in multiple population? It is like your content, or you took this from some paper, or something? Just to ask.
Oh, where did I get?
Are you sure that this view is correct, that you have shown us? No, sorry, of course you are sure if you are saying that.
So you're asking, where did I get the number for you?
Yes, no. I get, where do you think that development of a generation is, must be for sure like you are pointed, or you just, or it is just missed something in the maths?
Can you repeat the question, or you can even type it in the chat and I'll read it. Sometimes
nice.
I apologize to have to ask you that. I'm really sorry, but I really want to answer your question.
No, I want to just ask. So this is about this development of a generation. Is this your conclusion that this will be developing like that?
Oh, is that my conclusion? Or is that?
Yes, or it is from somewhere taken by its original, yes.
Oh, good question. So I'm just extrapolating. That's not specifically in Dan Graur's literature. That's not specifically in Dan Graur's literature, but I extrapolated that where he came up with that figure of, I'm sorry. Let me rewind the slideshow a little bit. I'm going to rewind it back. So you had where it continues to accumulate. So let me bring this up, Muller's limit. Thank you for your forbearance. Let me try to bring up the slide in question. And I'm just gonna, rather than put it in slideshow mode, I'll just show the PowerPoint here. It's gonna be easier for showing it. So do you see this? Is my screen showing this slide where it says 5 times 10 to the 35th?
Yes.
Okay, so.
Okay, no, it's.
So this is, I'm just gonna quote, this is what's in the literature. That other graph where I was showing how it accumulates over time, over many generations, that's my inference from the literature. I don't know that, my guess is that the authors presumed it would be so obvious that the mutations would accumulate unrestricted. And that corresponds to, let me see, Kondrashov. The idea of mutations accumulating over time was this one. This is Kondrashov's paper. Why have we not died 100 times over? And in that paper, if you read it, he proposes one solution, but he did say that it can accumulate mutations over time unless there's some mechanism to clear it out. He proposed a mechanism called synergistic epistasis. And I had a slide that, just for completeness, I said there was one solution proposed. It's not, I don't necessarily believe it's right, is this synergistic epistasis to remedy that problem. And then there's another one called soft selection. And I said that was,
okay. So can you explain it shortly what does this mean? Soft selection and synergistic epistasis.
I'm not familiar enough to do the topic justice. And I'll tell you why I decided not to be familiar enough with them. Dr. Graur here, who's also a respected evolutionary biologist, this calculation shows he doesn't believe that those mechanisms are valid.
Awesome.
So we will have some people that, I'll tell you what happened a little bit, because there are evolutionary biologists like Kondrashov who worked at the NIH. Let's see if I have a picture of Kondrashov. He worked at the NIH. I would guess that he thinks most of the genome is functional. And so he's trying to find a way that he can reconcile the high mutation rate with the functionality of the genome. So he came up with this theory of synergistic epistasis. The solution Graur proposes is that instead, let's just assume the DNA is 98% junk or some fraction, some large fraction. So is there resolution to this current impasse, to this conflict? There's none. We have faculty at the same university arguing over this topic, and it's not resolved. The creationists, somewhere the creationists slip in there, and they actually tend to side with Dan Graur, obviously, because he said if ENCODE is right, then evolution is wrong. And so when I was in a debate with Dr. Dan over genetic entropy, I cited this, and Dr. Dan said, well, Dan Graur is wrong. So that's one evolutionary biologist disagreeing with another evolutionary biologist. And the creationists will agree with Dan Graur. If ENCODE is right, then evolution is wrong. So my role in this, in the reporting of all this, was to study what the Encode Project was. And I'm happy to give a lecture on what Encode discovered and what they actually did. It's been misinterpreted. I've alluded to it in one of my discussions on the Encode Project and function in the genome. So I'm sorry, I can't give you like a direct answer as to who's right. I can only say what my opinion is, and I tend to side with Dan Graur. So there are people who would disagree with me. Evolutionary biologists would disagree with Dan Graur, but he's very respected up until recently. So please, question.
Okay, so You just wanted to tell something about ENCODE. So what?
Do you want to have another discussion on ENCODE? We could, because that's we've already gone an hour and my voice is running out. It's not that
okay.
I enjoy talking to you, but my voice is not cooperating, and I really appreciate you reaching out. I know you had a series of questions, but
yes.
I'm not saying that, I've tried to lay out, why I have the opinions I have, but I've today I've tried to lay out the historical, mostly the historical and the math, the development of junkDNA theory. So have you heard the term junkDNA used a lot?
Yeah.
It originated from all this mathematics. That's where the term junkDNA, and so Ohno used Muller's math, and he used the Muller's limit, and he concluded that's where he said most of the genome has to be junk based on Muller's limit.
Mm-hmm.
So that this is more of a historical treatment of the idea. I obviously have my beliefs on who is closer to the truth, but I hope at least I've laid out what the opinions are and the relevant papers for both you and everyone else listening to this talk. They can decide for themselves. And I hope the references I've laid out will also be informative. So students that really, people that are really interested in this topic can read the original papers. And I would welcome, if there is anything I didn't represent accurately as far as the peer-reviewed papers, I'm happy to get correction on that.
Yes, thanks. So if you feel like it's enough for today for you, that will be fine, or else I have some questions, but we can maybe make it next time.
Yes, well, why don't I list down the questions you have, and then that way I can prepare something like I did today. I have,
okay.
It was just nice I happened to have given this lecture earlier, and you've given me a chance to repeat it and make it actually even a little better than the last time I presented it. So, and I know you gave me a list of questions that you had in an email. So the first one was on Fisher's theorem, and then Basner and Sanford, and then now is Muller's limit in junkDNA. Do you want me to go back to our email on, or maybe you have a different set of questions, and that's fine too.
So I wanted to ask, how would you determine if the piece of genome functional or not functional? That would be a good question.
Okay, functional. That's actually a really good question. Just briefly, I think it's this is very interesting. It actually relates to my discussion with Dapper Dino on fitness. And first off, so this will be another topic and I have to think about it. But the first thing is I would not use the definition of evolutionary fitness to define function. So that's why I had that debate. I was going to say the way we determine it is determine function is based on engineering principles. So if someone doesn't accept that, I respect that. I can only give you my viewpoint. The engineering viewpoint is kind of adopted by the medical community. So like even though for example, even though Tay-Sachs disease and sickle cell anemia have been cited as beneficial mutations by some evolutionary biologists, in the medical sense, they think that's just terrible, that it's a disease. And so there's a dispute. Yeah, so that's why I was saying to Dapper Dino, I said, the engineering and medical community have a different view of what constitutes functionality. It's not exactly the same as the evolutionary biologists who tie reproductive fitness to the definition of functionality. So that's one reason I felt that debate was important, because what we call functional is not, people also disagree on that. And so I was trying to articulate how we might be able to do it and define function independent of reproductive success, and also independent of assuming a designer. We could probably do it if we just say, just like what I was trying to outline with the bird, we don't have to assume God created the bird, even though I believe that. We could just say, well, the bird has a magnetic compass, and it gives it the suitable equipment to be able to navigate 40,000 miles a year. And I was hoping that wouldn't be very much under dispute, that we can call that magnetic compass is a functional system. So the question is, how do we do this in terms of the genome? And I can articulate it from that vantage point if one is willing to at least admit, for the sake of argument, kind of the more engineering medical notion of functionality. I will not be able to argue it in terms of reproductive success. That's why I had a problem. It becomes really difficult to identify function if it's just defined in reproductive success. But if we can define it,
but roughly you can count this is the same. So for me, it's fine.
There are times we can, but there are also times when having function, carrying function, can be limiting reproductive success. And that's why I cited the example of very high IQ women being childless, having a high proportion of childlessness. Most people would not consider someone who's intelligent. So if we take two female individuals, they're all equal except one is smarter, we wouldn't view the smarter one as being defective. But if we use the reproductive sense, she would be considered having deleterious traits. And that's why I had to kind of just raise the issue. I said, there has to be a way we can evaluate function without having to couple it necessarily to reproductive success. And there are also other examples like this blind crustacean. I think things like crabs or crustaceans. There's one particular crustacean that is blind, and it has more reproductive success in a very dark environment than its fellow species that have eyes, because the species that have the creatures in that species that have eyes, but they're operating in a cave environment, they're having to use a lot more metabolic energy to sustain eyes that they don't use anyway. So therefore, selection will favor the blind crustacean. This was a topic that was raised by an evolutionary biologist named Alan Orr, and the name of the creature is called Gomeris minus. But an engineer will say, well, a functioning eye is still a functioning eye, whether it's reproductively successful or not. And so I can only kind of go, I can go to the engineering sense if you'd like me to explore that. I mean, it'd be a good way to learn some biology too.
Yes.
Okay.
But for example, creature would do not need eyes, like living cave or something. They might have other mechanism to look around, like bats have sound or per smell, like some other creature do that.
Yes, they'll be able to operate. Yeah, like bats. Bats are able to use their sonar in caves. So I can specifically talk about the functioning in the human genome and my work on that, and then I could talk about other creatures. So just from a standpoint of just learning what's in biology, without arguing creation or evolution, I just find it fascinating how biology works. To me, it's very fabulous. So we can talk about that sometime.
Okay, that's good.
All right. And then if other questions come up, we'll try to address those too. So
yes, I have last questions. So in the slides where you showed that amount of mutation is accumulating, so can you roughly say what amount of mutation can a human still hold without detectable effect, and what amount of mutation does cause effect on human? Just roughly, so you can just make the broad overview for the
Yes,
numbers.
so that's a very good question. No one knows the exact figure, but what we can say based on Kondrashov, and let me see if I could put up my slide. Oh, I already have my slideshow up. So Kondrashov in the crumbling genome, Kondrashov said, see if I have it. Do I have it, the figure he gave? I don't want to have to go through all my slides to find. He said we already have about 100 mutations. So the estimate is we have 20,000 genes, and then 100 of our genes, every individual has already lost maybe 100. So one person may lose, it may be
So
different sets of genes for different people. So I
you
would
mean
say we
100,
could,
you mean 100 broken genes?
100 broken genes.
Mm-hmm.
And I had that earlier, and I'm really sorry, I don't know where I have.
From 20,000 genes, okay.
It was somewhere here. I quoted it earlier, and I have so many slides I won't be able to find it, but it was in Kondrashov's book. So that didn't directly answer your question, but we're already surviving with broken genes. How many more we can afford to lose, I don't know. I will tell you this, bacteria, we know bacteria can lose maybe 80% of their genes, like the E. coli, and they'll still survive. But they're not, bacteria are not as complex and sensitive. So we've actually had, we have this thing called an immortal cell line where we took a cancer cell and we're able to keep it alive and it reproduces. It's never going to be a human being, but it doesn't need a lot of genes, obviously, to be working because it, doesn't need eyes or anything. So I'm sorry I can't give you a precise figure, but let me see. I
no, it is what, so roughly 100 broken genes from 20,000 genes. So just roughly this is the picture.
Yes, and I'm going to show you one gene that was really devastating. Just a single gene. It's called, okay, just a single gene. So it's not just the number, it's which one that one loses. So there is one called the nuclear Lamin. The nuclear Lamin, defects, this is from the textbook, from my cell biology textbook. It says, defects in a particular nuclear Lamin are associated with certain types of progeria, rare disorders that cause affected individuals to age prematurely. Children with progeria have wrinkled skin, lose their teeth and hair, and often develop severe cardiovascular disease by the time they reach their teens. And it's really sad. Many of them don't live very long. And this is someone, life expectancy is age 13. So if it's one particular gene that's very critical, the effect could be really, really bad. So it's not necessarily the number, it's which genes. And that was one example. So I think the question is a little bit more complicated because there are obviously genes we could probably do without and still survive, and life might not be so pleasant, but then there are others, just a little defect could be very devastating. I do know, because I have studied some of these heritable diseases, there is a gene called HEX-A, and if it's damaged, that's just one gene. If it's damaged with a frameshift mutation, it causes Tay-Sachs disease. So there are some diseases that are single gene diseases. They're just devastating, and that, progeria is one of them. So it's not quite so easy to say how many can we tolerate. It's probably more nuanced than that. It's like, which ones can we actually get rid of?
Yes, I just wanted roughly to have in this model.
Yeah.
Okay, so.
So I hope that was helpful.
Yes, that was very good. Thanks, Al.
And if you think of other questions, we'll talk later. And I'm going to stop the recording, and I'll stop the screen share. And let me see. I'm going to stop the recording, and we'll talk a little bit. So let me just say goodbye to the audience before you and I have our little backstage just conversation. I'd like to extend greetings to all of those who joined this presentation. And Peter and I, it's been a privilege. And thank you for coming out to this talk. And feel free to put comments and any corrections in the comment section of this video. So take care, and it's the Christmas season, so I wish you all a Merry Christmas and a Happy New Year. Take care. God bless you.
Yes, also.
Yes, Peter, if you wanted to say something.
Yes, also Merry Christmas to you.
Thank you.
Yes. So, and